12th Annual UREP Competition. Welcome our distinguished panel of judges to another day of exciting research projects by our talented students. Joining me in the judges panel are Dr. Hisham Saber, Director of Technical Team at QNRS, Dr. Dhruv Arora, Research and Development and Technology Manager at Shell Qatar Research and Technology Center, Dr. Amar Bukhari, Senior Manager, Energy and Environment, Dr. Mohammed Jarrar, Senior Manager, Health and Biomedical Sciences, Dr. Munir Tag, Senior Manager, ICT, Dr. Hatem Henni, Senior Manager, Social Sciences, Arts and Humanities. As you know, yesterday we saw 8 teams present their research projects related to environment and engineering along with ICT. Today, we will see another exciting research project related to biomedical and health and social sciences, arts and humanities. Without further ado, I would like to invite the first team is from Weill Cornell, Amina Shafiq and Dana Al-Ali, presenting the role of a human gut microbiota in autism spectrum disorders and inflammatory bowel disease. Please share your presentation now.
We have a problem with the...
We can hear you, Amina. Go ahead.
Okay. All right. Yes. 1 in 83 children in Qatar are diagnosed with an autism spectrum disorder. Autism
I think the voice, excuse me, the voice has some problem, I think.
Can you hear us? Hello?
Now,
Can
yes.
you hear me now?
Yeah, but you need to be close to the
very
mic.
Closer.
Yeah,
closer
whoever
to the
gonna
mic.
present has to be close to the mic, okay?
Yeah, we both have different mics. Can you hear me clearly now?
No, still, there is an echo.
What about now?
No.
Right.
Dana, can you start? Can you just, I want to try your voice. This.
Can you hear me now?
Yes.
Yeah, much better.
Yeah.
All right, I'll just start.
Yeah.
I'll start again.
Yeah, can you please go to the first slide? Yes, go ahead.
The slide? All right. Good morning, ladies and gentlemen. My name is Amina, and with me, my partner Dana, are going to talk about our project role of the gut microbiota in autism spectrum disorders and inflammatory bowel diseases. Our PI is Dr. Ghazlan Bendris. 1 in 83 children in Qatar have been diagnosed with an autism spectrum disorder, also known as ASD, and it has become Qatar's national health priority. In addition to some genetic factors, environmental factors and lifestyle are thought to play a more important role in the pathogenesis of this condition than previously thought. There is strong evidence to suggest that there is a bidirectional communication between the gut and the brain. And this mostly depends on the bacterial fermentation of dietary fibers and production of metabolites and hormones by the gut. And this can be disrupted in a dysbiotic state. Our study is the first pilot study conducted in the country, with the main objective being to analyze the feasibility of implementing a fusion clinical trial involving dietary intervention on autistic children in Qatar. If we want to run such a clinical intervention, we need to be able to answer 2 categories of questions. First, how is the awareness of the population in regards to the importance of the diet in managing autistic symptoms? And is the population ready to engage in clinical trials that modulate the gut microbiome? Secondly, we need to understand the microbial profiles. We need to optimize our methods of recruitment, stool sample collection, extraction, and sequencing, and have preliminary data which could confirm the microbial diversity shifts that were observed in other populations. For this, we used a combination of online questionnaires and seminars to address the awareness and readiness among health professionals and the general public. Writing literature reviews was also an important way for us to raise awareness among scientists. For the other leg of the study, we collected stool samples from 55 individuals, which were divided into 3 cohorts. One was the autism spectrum disorder and the attention deficit hyperactivity disorder group. The other group was the IBD and the IBS, which is inflammatory bowel disease and irritable bowel syndrome group. And the third was the healthy controls. We used Fisher Brand's stool collection kit, which was easy to use for the participants to provide us with their stool. We then extracted microbial DNA using the QI agents kits, and then these samples were sent to CD Genomics for next generation 16 sRNA and ITS sequencing, and phylogenetic analysis was performed. We then used this data to compute descriptive statistics and correlational analysis using R and SPSS. And these are the key findings from the surveys. If we look at the proportion of individuals with obsessive compulsive and anxiety disorders among each group, we observe that there's a higher proportion among the ASD, ADHD and the IBD, IBS group. This, again, supports the gut-brain interaction that we were hypothesizing. Second, if we were interested to analyze the diet type of the population, and among the 112 participants, most of them in the three groups have a diet which is rich in processed grain-based products. We also wanted to know about probiotics, as it is a way to correct dysbiosis. And we wanted to know if the participants were taking probiotics. And as you can see, more than half of the individuals that could potentially benefit from these are not actually taking them. 30%, 50% of the ASD, ADHD, and the IBD, IBS groups compared to the controls. In terms of awareness, we also wanted to know about the antibiotic exposure among the groups. And what we found, which was really important, is that 58% of the ASD, ADHD group and 38% of the IBD, IBS group had several courses of antibiotics during the first 3 years of life. This is something that should be monitored in future studies. In terms of awareness, among 79% of health professionals and 54% of the public were aware of the importance of gut microbes in pathogenesis of disease. And this is something we should increase. Finally, we cannot run a clinical trial if there is no willingness to engage in them. Here we can see that 90% of the participants reported to be ready to engage in dietary intervention, and 70% even to engage in fecal transplant, which is another way to modulate the gut microbiome. Interestingly, we noticed that health professionals seem to underestimate the willingness of the public to engage in such trials. Can you hear me? Hello? Can you hear me? Can someone tell me?
Yes. Yep.
Okay.
Go ahead.
So now we'll be moving on to the microbial profiling results. And we can see that our data confirms what was shown on other populations, which is the presence of specific biomarkers in the gut, with 29 operational taxonomic units for IBD and IBS and 25 for ASD and ADHD. Now, the originality of our study also comes in because we studied the microbiome of this, which is often neglected in other studies. And we can see that this was very fruitful because 1,638 taxonomic units were identified for ASD and ADHD, and 775 were identified for IBD and IBS. Now, the main key and expected finding for the study is the loss of biodiversity in the gut among the disease groups. And we can see the lowest among the ASD and the ADHD group. And now Qatar, for the first time, is confirming the same trends that have been shown in other populations. The decrease in biodiversity translates into the absence of millions of bacterial and fungal species in the autistic individuals. So we created the database that's consistent of 7 bacterial species and 106 fungal species that are all significantly less abundant in the autistic group in comparison to the controls. And we can see some of them displayed on the side, one of which is the Bifidobacterium longing, a very important bacterium in the gut, which is found 3.4 times less among the autistic group, further signifying that biodiversity in the gut is correlating with a healthy state. Samples of the ASD and the ADHD group did not only show decreased abundance, but they also had overgrowth of some bacterial and fungal species. We have samples of 2 right on the side from the order Clostridialis for bacteria and the Codnea order for fungi. Now, one of the key findings of this study is a fungi from the Willemia order, which was found 37.2 folds more abundant in autistic individuals in comparison to controls. Now, this species is unidentified and has not been reported previously in literature, highlighting the significance of this research and the significance of this finding for further investigation. Due to these findings, the loss of biodiversity has become an important therapeutic target
to treat autism, having launched many clinical trials across the world that aims at modulating the gut microbiome for the treatment of autism. There are two methods to do such a thing. One would be fecal transplant, and the second would be through dietary interventions and lifestyle changes. One clinical trial that published its results recently shows this results, where after 8 weeks of fecal transplant, we can say that the severe symptoms were reduced by half, and after 2 years, 40% of the autistic individuals had minimal to no symptoms. Now, of course, fecal transplant does not come without consequences and side effects, and hence we are proposing the use of dietary interventions as a less invasive, more conservative method. However, it will take a longer period of time for it to modulate the gut microbiome. So to wrap up, ladies and gentlemen, we can say that we have collected a set of very important data that allows us to say two important things. One, the awareness and readiness of the population is favorable for the proposition of such a clinical intervention. And we did this by using 3 reviews, 1 that was published, 2 pending publication, a seminar, a grand round, and 2 poster presentations. We also proved to you, ladies and gentlemen, that there is indeed a need to reevaluate the diet that autistic individuals have, specifically focusing on the processed grain-based products by exchanging them into fibrous foods and also introducing probiotics into the diets of these individuals. The study has some limitations, including a small sample size of 55. However, we'd like to reassure you that everything was well documented and supported. Similar to any other studies, the microbial profiles are very difficult to generalize because of the difference they can create on the composition in terms of population, techniques, and diet, all of which can affect the composition. Looking into the metabolome is also something that needs to be considered for future studies instead of just taxonomic data. Finally, the project is not only ending its step here, but have yielded 2 different projects looking into the probiotic formula for ASD and other methods to release anxiety, all that surfaced because of the results of this study. Finally, we are now in possession of an important set of data and the network of collaborators from CIDRA, HMC, the Child Development Centers, and Weill Cornell University that enables us to propose a clinical intervention on autistic children that aims at modulating the gut microbiome by involving a complete lifestyle change. And this would be the first clinical trial available for autism in Qatar and in the Middle East region, making Qatar a pioneer for such research in the Middle East. We would like to acknowledge QNRS for the fund, as well as Ibrahim Leswi and the pre-medical department at Cornell. Thank you very much, and we're open for your questions.
Thank you, Amina, and thank you, Dana, for your very informative and clear presentation. I may ask now Dr. Mohammed Jarrar to raise his first question.
Yeah, thank you, Amina and Dina, for this nice presentation. I have a couple of questions and some notes to raise for you. I know this is a very complex type of research, and it has been going for all over the world, actually. Many people interested in this and the implication of microbial biodiversity in so many health disorders, including metabolic disorders and ASD and IPD, as you mentioned. Now, I have one of the limitations you mentioned, or you forgot to mention in your study, is that the cohort, the homogeneity of the cohort in your study is a very important issue here. What is the age of your subjects? Are they taking medications or not? Usually you look at the demographic area and type of food and diet they take. All of these are factors that influence the microbiota in evaluation and also in the intervention in the future if you think to go ahead and design an intervention therapy. So just to check, one of the questions, can you explain for me from physiological and biological point of view how the dysbiosis in ASD patient may alter the gut permeability And so the behavioral changes in those ASD patients and IBD patients.
Yes, so you are absolutely right. There are many factors that affect the gut microbiome. And that's why when we did our questionnaire,
excuse me, we can't hear your voice.
yeah, what Dr. Gerard mentioned is 100% true. There are many factors that affect the gut microbiome. And so when we did our questionnaires, we had an extensive list of questionnaires: the diet, the ethnic and nationality, their diet type, probiotic use, antibiotic. We have almost 26 variables that we took into account in our questionnaires, and it's a very extensive analysis. We're still going to do the correlational analysis on it, and these results are still pending because we're still working on them. But we tried to take as much variables as possible into account because we know that everything affects gut microbiome, and we wanted to make our associations more accurate. As for the effect on the gut permeability, so the microbiota composition of the gut, how they affect the human body is through the secretion of metabolites like butyrate, propionate, acetate, and all of these alter the gut's permeability by affecting the tight junction expression and the secretion of mucus. For instance, butyrate, when there's excess of butyrate not excess, when there's an appropriate amount of butyrate, it increases the tight junction expression and it increases mucus production. This again decreases the gut permeability, so less of the shorty chain fatty acids and the metabolites are able to escape outside the gut and induce negative impacts like triggering immune reactions and neuropeptides like serotonin. Serotonin is found to be very important in the pathogenesis of autism. 90% of serotonin is actually produced from the gut. And it's very easy to understand like how this could potentially affect the brain. And that is why we took this as our project, because we know that there's such a strong connection between the gut and the brain and every other organ, to be completely honest.
That's very good. Plus the pro-inflammatory cytokines, remember these in the IPDs, they play important role. The last question, quick question, the interesting in your project is that you're finding about this Basidiomycota fungi, which is very high in the ASD patients. Did you look into what role this fungi could do in this metabolites productions and how that could influence the behavior and the health of those patients?
So we are the first ones to have identified this particular species as being that much increased in our population. So we haven't been able to find papers or research on it, but we want to work on it if we have a larger cohort, and if we can do, our future plan is to focus on a metabolic analysis instead of a taxonomic analysis, so we can understand how exactly they play a role in the pathogenesis of disease.
Okay, thank you.
Thank you. I just have one question. You know that you're talking about the relationship between the autism and the microbiota. So what happened in the absence of microbiota during early life? So what will happen if there is an absence of microbiota?
So basically what our study has found is that the first 3 life is the stages where the brain and the gut are actually developing. So the brain and the gut are among the organs that even after we are born, they are still continuously developing. And the first 3 years of life is where they start maturing and developing even further. And studies have shown that with the decrease of microbiota among these stages and early exposure of antibiotics, there's further correlation with having autistic symptoms, ADHD, and finding other disturbances in the neurological aspects of these individuals. In fact, one of the bacteria that we have mentioned, the Bifidobacterium longum, when it was found in decreased levels, when it was found in higher levels, these individuals were seen to have less childhood disorders in comparison to individuals who did not have bifidobacterium longum in their bacteria. So by exposure of antibiotics and decreasing the biodiversity among these individuals, you are predisposing individuals to having neurological symptoms such as autism or even conduct disorders in these individuals.
Thank you, Dana. The next, I think, Dr. Hisham Saber. Dr. Hisham.
Yes, thank you, Doctora. Thanks, ladies, for the very interesting presentation. I'm not of the field, so you'll have to excuse my ignorance of it. Your hypothesis is that there is a causal link between the microbiota, gut microbiota, and ASD. But the underlying suggestion or hypothesis is that it is the cause of ASD. Is there a prospect that it being the effect rather than the cause, that it is caused by ASD rather than the other way around? I know the literature that you showed improvement of the situation by altering or amending the microbiota, which supports your initial hypothesis. But is there a prospect or a chance of it being an effect rather than a cause of ASD? Thank you.
So we do agree that there is genetic basis to the disease. There is some, but as you can see, there's
your voice is very far. We can't hear you.
So I'll take the question. So yes, this could be a possibility, and this is mainly because when we're looking into the studies for autism and as well as ADHD, one of the most important comorbidities that children with autism have is GI disturbances. And when you have GI disturbances, so these, for example, children did not like, for example, to eat certain dairy products and other things which are considered probiotics for your gut. So it could potentially be a cause. Right now, we are establishing that there is, in fact, a correlation between dysbiosis and an autistic state. We cannot say that it is a cause. It is merely correlation at this point. However, a lot of studies have been showing that GI disturbances being a comorbidity in ADHD and ASD children could be an indication that there is a pathogenic link in it, or it could be, as you said, the fact that because they have this GI disturbances, they are not eating probiotics, causing this loss of biodiversity and reversal and hence. And there's also a study that was published recently by Nature that showed that autistic symptoms can actually be reversed. So the study was conducted on mice, and they found that after giving these mice probiotics, they found that there was reversal of these autistic symptoms. Also, a documentary that was recently we've recently watched on autism also showed that they interviewed mothers, and they showed that when the mothers started introducing probiotics into their children's diet, there was a massive improvement in their autistic symptoms.
Thank you very much. You answered that amply.
Thank you. I think we run out of time. Thank you, Amina, and thank you, Dana, for a very interesting presentation. So the next up, we have Lubna Zarif from Qatar University to present the research titled Effect of Crude Extract from Qatari Medicinal Plants and Breast Cancer Cell Proliferation.
Hello, good morning, everyone. This is me, Lubna, I'm a student at Qatar University, and I will be talking about this project that we did, and it was called Effect of Crude Extract from Qatari Medicinal Plants on Breast Cancer Cell Proliferation. So at first, I would just like to say that breast cancer is one of the most common type of cancer that's affecting women worldwide, and it's one of the second type of cancer that is affected in Qatar, both residents and citizens, and it's 39%, and the survival rate is 58% in late stages of diagnosis. So our study included the use of two Qatari medicinal plants, convolvulus and Plantigo ciliata, to find their effect on breast cancer cell line that was MDA MB231. So the reason why we chose these two plants are because previously they have shown anti-inflammatory and antiseptic properties, but their effects on any kind of cancer cell is not explored. So we were very interested to look further into that. So there were three objectives of the study. The first was that we were required to prepare the crude extract from both these medicinal plants and using three different solvents, which was water, acetone, and methanol. And the second objective was that after we extracted the crude, we wanted to test its effect, anti-proliferative effects on, as I mentioned, the breast cancer cell line that we used. And the third objective was to determine which was the best solvent out of the three that we used. So I will be explaining the methods of, in brief for the crude extraction process. The first series of pictures that you see over here are for acetone and methanol. Basically, the procedure was the same for both. What we did was first we started with plant preparation, which was we kept the plants for freeze drying for about 2 to 3 days, and then we grinded them. And after grinding them, we used the powder for the extraction, where we dissolved it in acetone or methanol, respectively, in like separate containers. And then we use nitrogen gas for once the extract was dry, to get the dry extract, we use nitrogen gas. And then came the third step, which is the crude dissolution. We used DMSO plus media to dissolve the dry extract that we had obtained. The second picture that you see here is us using nitrogen gas for evaporation, and this is the final, this is the dry extract that we had obtained, and here is the picture for dissolution that once we had added DMSO and media to it. For water, the process was a bit different. The plant preparation step was the same. We freeze dried them, and then we grinded the leaves and the stem of the plant, and we boiled it. And after boiling, we evaporated, we kept it in a Petri dish, and then we let the evaporation step go on. And then we dissolved the dry extract, which is here that you see in the Petri dish. This is dry extract. We dissolved it again in DMSO and parts of the media that we were using. And then coming to the next step was the cell viability assay. We went for LMR blue assay, which basically works. It's a dye. LMR blue is a dye that indicates the fluorescence. Like basically it changes the color depending on the redox reaction that takes place once you add the treatment to the cells. And this equation that we used, this was the equation that we used to calculate the percentage yield in order to know how much was which solvent had a better yield. So this is the equation that we used. Going into the results for Plantago ciliata, as you can see over here, there is a significant decrease. The blue is an indicator of water. The solvent that we used was water. There's a significant decrease in the cells that were treated with the water solvent, having the crude extract. So here, as you see, the color is changing. So this is the control. So A and B are both acetone and, A is methanol extract and B is acetone extract. And you can see that there's a decrease. And here the cells, it's dark blue color, which means that there are more dead cells. So the darker the color, that's the indicator. The blue color indicates the dead cells. So the more the blue color, the more dead cells you have. For acetone, you see that there are some in 2 milligram per milliliter, you see a certain change in color, and even for 60 milligram per milliliter. But for the water extract, you will see the difference in 6 milligram per milliliter, which is a lot more. Like you see the difference, you see a significant difference in water than you see in acetone and methanol, and I'll explain why. Also, you can, we were able to get the IC50 range, which is 50% of the reduction of the breast cancer cell line in just this concentration within this range. Okay, and this was for Plantago ciliata. For convolvulus, we got similar result even though we got a better result for Plantigo ciliata, we found the same trend. But over here, as you can see, there is not much reduction. So acetone and both methanol extract of convolvulus did not have a very significant effect. But we can see that kind of effect in the water extract around the same range, 6 milligram per milliliter to 15 milligram per milliliter. There was a 50% reduction of the breast cancer cells. So I would like to say what we found in terms of solvent was that water is found to be having the highest yield when we calculated, which was around 58%, as compared to acetone. We only had a percentage of 2% of extraction yield using acetone. We explain it due to high polarity reasons, like high favorable conditions like high polarity, temperature, and shorter extraction time. As you know, water is a more polar solvent than compared to methanol and acetone. This is to do with how it binds with the active ingredients in the plants. The more polar the solvent is, higher the extraction yield. That's why in acetone, even in absolute form, we were not able to extract perhaps all active compounds present in the plant, and that's why it had the lowest recovery. Previous studies support this our findings, because water extracts in many studies have shown, first of all, cytotoxic effects on various cancer cell lines, plus they have shown to be a solvent for a very high extraction yield. This was a study that we found, but it's another species, Plantigo major. Hot water extracts from the leaves that they used had shown high cytotoxic and anti-tumor properties when tested on prostate cancer cells. So in our plants, what the compounds that are perhaps the active ingredients are steroids, alkaloids, and terpenoids. And in convolvulus, we have alkanoids, flavonoids, and sterols. But we did have a lot of limitations in this study because of time constraint. What we generated is a very preliminary data.
Excuse me, Lubna, you have 2 minutes left.
Okay. We were not able to identify the specific active ingredient in the plants that caused the cytotoxic effect that we observed. And also we only used one cell line, so it would have been better if we were able to use, it would be perhaps more necessary if we would have used other cell lines, cancer cell lines, to test the anti-proliferative effects. In conclusion, I would just like to say that this project has, for future, it has a very wide spectrum. We can use different cell lines, we can identify different pathways involved in apoptosis, and we need to identify the bioactive compounds that mediate the cell inhibition. And also this project highlights diversity of Qatari medicinal plants that are not explored yet and can be explored. Thank you. I would like to thank QNRS for funding this project, and also Qatar University and Anti-Doping Lab for providing the available materials and equipment for research. Thank you. I'm open to questions now.
Thank you, Lubna. The first question from Dr. Jarrar, please. Go ahead.
Thank you, Lubna. Thank you for this research. So from your finding, I understand that you saying that the water extracts for these plants are more effective in killing these cancer cells, okay? And the ethanol and acetone is not effective as water extracts. That's okay, this is good finding. In other words, you mean that plant extracts are organic compounds, some of them water soluble and some of them soluble in organic solvents like acetone and ethanol. So in other words, you're saying that whatever that extract, whatever that organic compound that has effective on these cells is water soluble, yes?
Yes.
So that's the interpretation, which is okay, and it's important. But
yeah.
Now, my question is, did you use controls? What other control? Did you culture cells only on water without water extracts and you compare it to that? That's one thing. And also the ethanol and methanol itself wouldn't have an effect on these cells. So yeah, this slide. So
Yeah.
you use water only?
No, so basically in control, we had the cells and we did not have any,
Any
we just,
extract?
yeah, we did not have any extracts.
So water only,
So it's
water
just
plus
cells with,
extract.
yes,
Yeah.
and yeah.
Okay, good. So the question for you is why you use DMSO? That's one question. The second question, I know you evaluated the viability by Elmar Blue dye, which is okay, it's routinely used. But did you try to look at the cells under microscope and try to count them and see if that correlates with the number of cells that you see compared to the bluish color of that cells?
Yes, for sure. Because first, I'll answer the first part of your question. We use DMSO because we had troubles dissolving the dry extract with just media. So we use very less quantity of DMSO just for the complete dissolution of the extract, because if it's not dissolved, our results would not be accurate. And second thing, yes, we did count them under the microscope. We actually did this several times to achieve this, because we did not have an optimized concentration or an optimized procedure for this. So basically, we were just trying what works at first. So we did undergo the step where we counted the cells and we looked if it's actually causing a significant decrease, rather than just looking at the color change of color, and we found that there is a decrease.
That's good. So one more control you need is to include healthy cells, not cancer cells, the MDA cells, because
Yes.
you never know, maybe water with this extract will affect the viability of any cells. So you end up
Yes,
with no value of that, huh?
yes, we have this limitation that we should have used a normal cell line with a cancer cell line. As a research assistant now, I see the limitations of the study, but I also see that it is capable of a lot more that is not explored yet. I am working in cancer research now, and there are many possibilities of this project that can be explored and are yet to be explored.
Okay, thank you.
Thank you.
Thank you. The next is from Dr. Amar.
Good morning, Ms. Lubna. Thanks for the presentation. My question is about the relevance of your results. Where and how can we make use of your results? Can you discuss this, please?
We can definitely, if we have a preliminary data now that we have generated. So where we can use is, we have this plant that is probably very, very useful in cancer research that is not yet explored. So what we have generated is a fundamental to build something upon it. I think that's the relevance of this study, just to generate a preliminary data and then build upon it. In future, maybe these two plants that we have found can be actually a very good source for the inhibition of various types of cancer. Right now, what we have is just for breast cancer, and we have a very basic data, but with proper equipment and with more intensive research, I think we can take this study to a next level. We can identify various pathways like apoptosis, what actually goes on with the inhibition of these the different types of cancer cell lines. I hope that I answer your question.
Yeah, thank you.
Yes,
I think really next step, you need to focus on the apoptosis of the cell proliferation,
yes, for sure.
especially using the medicinal local. I think these are local, right?
Yes, they're local.
Yeah. So who's next? I have received some. questions. Dr. Munir, please.
Yeah, good morning. Thank you very much for the interesting presentation. Yeah, I mean, I think you have quite interesting findings. There are also some studies in the region and in Jordan using such type of plants.
Yes.
Maybe it would be interesting to look at this.
Yeah.
And also, you have also a long way to go. I mean, you have to check also the toxicity of these plants, you know, and also the side effects in the future. So
yes,
a long way to go, but it's quite a promising finding. Thank you. Just an observation. But can you comment on that, on the way forward?
Yes, for sure. As I mentioned, that we have to use. There are so many things that we could not identify actually, and we can build upon it. And those are the underlying mechanisms like mediating cell inhibition and different kinds of like I mentioned, apoptosis. That's a very important thing, that very important experiment that needs to be conducted. Various types of antibodies that need to be. What are the antibodies that are being inhibited? All these are things that I think we need to find.
Thank you.
Thank you so much.
I think also you may need to study the phosphorylation level of the extracellular signals.
Yes, and yeah. Also we were not we could not pinpoint what exactly is the active compound in the plant that is causing the possible inhibition. So this is also a part that we need to study and look forward to.
Anyway, thank you very much. This is a big project, and that was my specialization when I did my PhD. So thank you.
Thank you so much.
Thank you, Libna. Thanks. I think now we are moving to the social science, arts and humanities pillar. I will invite Yusra Hamana and Aisha Al-Thani from Qatar University to present their research that explores the impact of ICT's investments and urbanization level on environmental quality in Qatar. Welcome both.
Thank you. Good morning, everyone. Today, I and Ms. Aisha Al-Thani will present our project. It's done by us and our colleague Asma Al-Maadid under the supervision of Dr. Lama Sharfuddin. The topic is about the impact of ICTs and urbanization on environmental quality in Qatar. I want to mention that the statements made here are totally under our responsibilities, and nothing and no views expressed here reflect the Qatar University QNRF or their staff. We will present for you the background, methods, result and discussion, policy implication, and the conclusion. I want to give you first background information. We all know that air pollution is a big challenge in worldwide, but precisely in Qatar, we found that air pollution is a challenge despite the huge efforts in macro and macroeconomics level to improve environmental quality. And this is one of the main reasons that motivate us to investigate about how to improve environmental quality in Qatar. However, Qatar is ranked in the top 2 countries in terms of CO2 emission and the first in the ecological footprint. And this is because of the rapid increase in population and the energy consumption because of several important future deadlines, including Qatar National Vision 2030 and the World Cup 2022. The environmental quality is very important for the state of Qatar, and they are trying to support it, especially in the QMV 2030. We found that environmental sustainability is addressed in 3 out of 4 pillars of their improvement. Also, there is a large number of initiatives that the country have been implemented, like new technologies, smart urbanization, and smart cities like Lusail City. Our goal is to investigate the Our goal is to investigate the effect of information communication technology and urbanization on the Qatar environmental quality and propose recommendation to improve the environment. The objective is to answer the following question: can ICTs and urbanization improve the quality of the environment in Qatar? Does the environmental Kuznets curve hypothesis hold for the case of Qatar? And which energy and environmental policies can be recommended to improve environmental quality in Qatar? I can't move the slide. Okay. Moving to the literature review, we have 3 theories for each relationship. Each theory have a different impact on the environmental degradation. For the urbanization environmental degradation nexus, we have the urban environmental transition theory, the compact city theory, and the ecological modernization theory. For the ICTs and environmental degradation nexus, we have first order effect, the second order effect, and the rebound effect. For both relationships, we have a very mixed results based on the on different factors, like the group of countries we have used, the sample period, the proxy of environmental degradations, the econometric methods we have employed. Moving to data sources and variable definitions, our dependent variable is the environmental degradation. It has 3 proxies: the ecological footprint, the carbon footprint, and the CO2 emission. Our independent variables are as follows: the real GDP per capita, we used ICTs, urbanization, and also the energy use. The data set is covered from 1975 to 2016 for annual data, and from 1990 to 2016 for quarterly data. Our econometric model is a stirred-path model. It's a reformulation of the IPAT model. Here, the I is the impact and P is the population. To make it more simple, we add a logarithm and more variables. So we came up with the final model where beta 1 and beta 2 measure the impact of GDP on environmental degradation, and also inform us if we have an environmental Kismet curve hypothesis for the case of Qatar or not. Beta 3 measure the impact of energy use on environmental degradation. Beta 4 is the impact of ICTs on environmental degradation, and beta 5 is the impact of urbanization on environmental degradation. And finally, we have the error disturbance. The empirical methodology can be summarized in the following steps. First, we determine the degree of integration for all variables. And second, we test for the long-run relationship using co-integration technique. And finally, we estimate the long-run and short-run relationship, and we check the residual stabilities. Now we will move with my colleague Aisha. She will give us the empirical results. Thank you.
Thank you, Isra. The following graphs describe the evolution of dependent and independent variables used in our study. I will highlight one of our main observations to spend more time on our model estimation results. In the first figure, we can see the evolution of environmental degradation proxies, where we noticed that after the discovering of oil and gas in Qatar, the level of pollution highly increased. The next slide shows the table of our correlation analysis, where the results indicated that ICTs improve the environmental quality for both CO2 emission and carbon footprint proxies, but destroys it in the case of ecological footprint. Regarding urbanization, there was a mixed result with different level of significance. Moving to our estimation model result, which is more interesting, the results showed that, we can see over here, there is evidence of an inverted U-shaped relationship between real GDP and the 2 proxies, CO2 emission and carbon footprint. As we can see, the first coefficient is positive and the second one is negative, meaning that EKC hypothesis holds in the case of these 2 proxies. Whereas in the case of ecological footprint, we can see there is a U-shaped relationship. The first coefficient is negative and the second coefficient is positive. EKC hypothesis does not hold in the case of ecological footprint. When looking at energy usage results, we can see here that all of the proxies are significant and positive, which indicates that energy consumption destroys the environment. As for ICTs, we can see negative and significant results for both CO2 and carbon footprint over here and here, which means that it improves the environment. However, for ecological footprint, it destroys it because the results are positive and significant. Lastly, for urbanization, the results indicate that it harms the environment for both CO2 emission and carbon footprint. As you can see here, it is positive and significant. Whereas for ecological footprint, it was not significant, so there is no impact on the environment. These two slides summarize what I just explained. Based on our results, we recommend the following points. We recommend continuing investing in ICTs as we saw that the second order effects hold. We also advise accelerating the effect of ICTs through encouraging consumers to use them. We recommend increasing the awareness of ICT's social negative effect to avoid the problem associated with the rebound effect. Finally, we advise promoting research and development in new technologies. In conclusion, the results indicate that ICTs can be used to lower the levels of CO2 emission and carbon footprint, but degrades the level of ecological footprint. We also find that urbanization's impact on the environment depends on the environmental degradation proxy used. As for our post-project plan, we are planning to finalize and submit.
Aisha, excuse me, you have only 2 minutes left or 1.5 minutes left.
Okay. So as for our post-project plan, we are planning to finalize and submit our in-progress paper for publication, aiming for a high-ranked journal such as Energy Economics and Energy Policies. Before finishing, I would like to highlight some of our project's achievements. We have participated in Qatar Green Building International Conference in the year 2018. We also published a research article in QNRS Research magazine. Moreover, 2 of our team members have been interviewed by Oryx FM Radio, where they discussed our project's outcomes. We would like to thank QNRS for their continuous support. Thank you. Yes, any questions?
Yes, thank you. I think I will start with Dr. Hatem, the first question.
Thank you, Yusra and Aisha, for this quite detailed and clear presentation, clear at least for me. I have a couple of questions. The first one is, could you remind me what are the environmental Kuznets curve hypotheses, please?
Okay, for EKC hypothesis, it states that when a country starts growing, it will start polluting, so the environment will degrade. After reaching a certain point of development, they will pour some of their incomes to improve the quality of their country. So afterwards, the environmental degradation will decrease in that country.
Okay, what are the 3 determinants of this evolution?
The income of the country. That's a really good question. I need to advise, but the technology, but I'm sure it's income also.
Yeah. Okay, does it matter? Okay. Now, if the EKC hypothesis holds, what type of policy should we recommend?
If the EKC hypothesis holds,
inverted U-shaped curve for Qatar, for example, what type of policy recommendation you will propose?
I think I'll propose about investing in ICTs.
No, if you have only as a variable the growth or the level of GDP, and only that. What would you say?
I'm not sure, but I'll look into it before publishing our paper. It's an interesting question.
Okay. I think you find an interesting result, which is the importance of the ICT. But at the same time, I didn't understand how ICT could improve environment. I know statistically your regression are really good and solid, but economically and as a behavior, how do you can explain this result? How using laptops and internet and mobile could improve our environment?
It's not only the use of laptops or phones. For example, for the case of Lusail City, using ICT here, it may be like a waste disposal system. They are using now waste disposal system, intelligent street lighting. They are using remote home automation to improve the quality of the environment. So through these steps, the state of Qatar is improving through
yeah, for sure, but
intelligent cities using ICTs.
Okay. Last question. I saw I think in your model you use a dummy variable, no?
Yes,
dummy
this
variable,
one.
the last one in 1998.
Yeah.
How did you choose this 1998? Is it ad hoc or there is something that
we
happened
used
here, which
you see? Yeah. This is a date of a structure break in 1998, where it takes the value of 0 before 1998 and 1 after that year. And we think maybe it happened because of the Kyoto Protocol that mandated industrialized nations to cut their greenhouse gas emissions at a time when the threat of global warming was growing rapidly. It was adopted in 1997.
Okay, so the impact is 1 year after.
Yes.
Okay.
Thank you, Dr. Hatem. Can we move, because I have 3 more panelists. The next one is Dr. Dhruv. Please go ahead.
Thank you, Yusra, and thank you, Aisha. Excellent presentation, and congratulations on completing this study. I have 2 short questions. The first one being, were there any non-intuitive findings that you really had from this project? Any revelations that you got? And the second one is just to know, can you define what the p-value is? The numbers that I see are 7.6 or ranging from 0.9 to something. So what is the definition of p-value? Let's start with the first question. What is the revelations that you got from this project, which were non-intuitive?
I didn't understand the first question.
So like Dr. Hatem was saying.
Yeah, answer the second question, then we'll go back to the first question.
Okay, let's answer the second question, the definition of p-value.
The first one was about student test, I think. T-stat. You understand me?
And can you repeat?
The question of my colleague, Dhruv, is about the T-stat in this slide. The T-stat is a student test, no? This is to say that your variable, which is, for example, GDP, is significant or not.
Oh, you mean if it's significant or not? You see,
yes,
for the three stars means that it's 1% significant. The 2 is 5%, and the 1 over here is 10%. Is this the answer you've been looking for?
That's okay. So maybe we can go back to the first question, which was a discussion that Dr. Hatem was having. The results that you got on dependence or how ICT actually improves the environment, that seems to be very intuitive in hindsight. But were there any revelations, that an aha moment that you had while doing this project and thinking, okay, this is something very interesting? All right, maybe we can move on to the next question.
I think the last question we will receive from Dr. Munir. Dr. Munir, go ahead.
Yes, hello, thank you for the presentation. Your conclusion has two contradictory outcomes. One, that the ICT is positive for the environment, but negative for the ecology, was what you said. Can you elaborate on that, please?
Yes. So after we did our estimation model test for ICTs here, log mobile users, we found that here it is negative and significant.
I mean, can you give me concrete examples in practice? I mean, beyond the formula, I mean, beyond the result of this analysis, concrete examples in practice. I mean, how ICT is positive for the environment, but negative for the ecology.
Maybe in case of CO2 emissions, like instead of going to work using the car, we can use the video meetings. So we will lower the CO2 emissions. But for ecological footprint, it takes into consideration 3 pollutants, which is air and land and water. Yeah, so we think it will have less effect. It is more globally here than these two parts, because both of them are air pollutant proxies.
Yeah. You are considering only mobile subscription, but ICT is much larger than mobile.
Dr. Munir, we run out of time. It's almost like a minute. Can we stop here and move to? Okay. Thank you very much. Thanks, Dr. Munir. Thank you, Yusra and Aisha. Is Sarah ready now, or shall we continue with the social science?
Yeah, can you hear me now clearly?
Can you talk more, please?
Okay. Okay, I tried switching laptops, so I don't know.
No, still you have the problem. No.
I don't know what to do. I'm sorry.
Wael, can we continue with the next presentation from social science?
The second one is
the second. Yeah, Sarah Al-Marri, Ala Dalloul, and Amna Sheep.
Ala is here.
Shall we start? Yes, okay. Please welcome with me Sarah Al-Marri, Ala Dalloul, and Amna Sheep to present, and this is a project is coming from Qatar University, to present the research with which studies the relationship between ICT use, including after-work hour emails and employees, work-life balance, stress, and occupational health, and exploratory study in the higher education sector in Qatar.
Thank you.
Yes, Ala.
Hello, everyone. My name is Ala, and I'll be presenting for you today a presentation which shows the relationship between the ICT use, employer and employer job satisfaction, work-life balance, personal burnout, and intention to leave. This study was done by a group of undergraduate research, including me and my colleagues, and it was under the supervision of Dr. Shada Abaid and Dr. Alam Abu Farha. I'll be presenting on behalf of my colleagues for today. To start with, to understand what is the ICTU as how we can see it nowadays, is that due to the technological changes
that happened nowadays. Everyone is depending on the ICT use to achieve their daily tasks and duties. Despite the positive effect of it on individual, there's still some limitation on the negative aspect. And this limitation can be explained by 3 reasons. The first one is that there is a few attempts of this field were specific to certain cases. And what do we mean hereby is that most research or articles will be focusing on a specific organization, cases of affecting the ICT use or specific variable. There were not a very big comprehensive model that cover all the details. And that leads us to the second point, when there is a missing point where there is a comprehensive model that examine the effect of the after working hours on the employer-related outcome. And lastly, we can see that most of the study were measured in developed economy, but none of them examined the developed economy such as in Qatar. And due to these 3 reasons, our paper and our research will try to investigate and develop a comprehensive model that shows the effect of the ICT use, which means the use of technologies such as emails, laptops, phone calls, on the ICT demand, which means the expectation of the employee that they have to respond to the use of ICT use and try to be able to connect to it 24/7. How these 2 relationships will result on affecting the employer-related outcome, which is the work-life balance, personal burnout, job satisfaction, and intentionally. To brief you about the method we use and how we found this comprehensive model is that we use a structured survey method, which is basically a survey that were distributed by my research colleagues in Qatar University during the May and November, using the method of a drop and pick up method, which basically when they will give the employee the survey, and then they were collected after 2 days. Once the data was collected, we used the smart PL system to analyze our data using 2 procedures, which is the first one, the measurement model that examined the relationship between the indicators and the variables. And the second model is a structural model. We show the relationship between the variables themselves. To talk about the result, we can see from this table using the recommended indicators of the PLS, we can see from the indicator loading that we tried to take all the data that were above 0.6 and removed all the data that were below to ensure the reliability of our data. In addition to the composite reliability and the core-band alpha, where we took all the data that crossed the cut line of 0.7, and the average variance, we took all the data that crossed line the 0.5. And this indicates that our data is reliable and can be used, which was distributed on the survey. The second table, which shows the validity of our data, which is a landed variable coloration, we can see from the diagonal line that it's positive and higher than all the data that were below. And this shows that our data is reliable and acceptable and can be used in the research. For the last table, where we use the R-square, Q-square, and the capabilities, it was recommended by many previous researchers to show also the validity of the data. We can see that the R-square ranges from 0.06 to 0.3. The Q-square also ranges from 0.04 to 0.2, which is the ranges that are acceptable, and it should be for between 0 till 1. And the capabilities as well from 0.06 to 0.8. In addition to the F-square, that ranges between 0.07 and 0.5. All of these 3 indicators show that our data is explanatory, powerful, and predictable, and can be applied to show the result that we are trying to have a comprehensive model as all. To show you the final result of the data, we can understand from this result is that there is a relationship between the ICT use and ICT demand and influenced by 0.2. In addition, how the ICT use will affect the 4 variable, which is the job satisfaction, personal burnout, and work-life balance. As we can see that the ICT demand affecting the job satisfaction negatively by 0.3, positively on the personal burnout by 0.5, negatively 0.2 on the work-life balance, and 0.5 positively on the intentionally. To discuss them more, we can conclude from the hypothesis that were founded within our research is that the ICT use is significantly and positively influence the ICT demand. The more use I'm putting an employer to use the ICT, the more he feels that he needs to be connected to it and try to adapt to it. Secondly, is that ICT use significantly and negatively influence the job satisfaction. The more demand on them, the less satisfied. thirdly, is that the ICT demand is significantly and positively influence the personal burnout. The more demand I'm putting on employer, the more they feel frustrated and anxiety that they have to adapt to this technology. Fourthly, is that it's negatively and affecting the work-life balance. The more ICT demand on them, the more they are unable to balance their life, to feel that they have to be connected 24/7 to the ICT and to reply to all the emails, phone calls they have. And lastly, is that the ICT is positively influenced the employee intentionally thinking that once I have a lot of demand, the employee will believe that this place is not suitable for them, and they believe that they need to leave so they can escape the ICT. Despite these hypotheses that we confirmed at the comprehensive model, there were some study limitations we believe as our researcher will face, is that the first one in our study, we try to conclude association, not the causation. So we focus on seeing the relationship between the ICT use and the ICT demand, and how then the ICT demand will affect the 4 variable, not the cause of those relationships. The second thing is that this paper we're focused on the theorizing and building the theory rather than testing. So we did not testing a specific thing or a specific hypothesis. We focused on building the theory and try to ensure that this theory is accurate, and it can be done by different method. The last thing is that our paper was restricted, which is basically done in Qatar in a small group of people, and we cannot generalize it as well for our findings. Despite those limitations, we believe that our project can contribute to many organizations and many places. And on theory perspective and contribution, we believe our paper will help the HR theory to understand how the model of the negative effect of the ICT use can affect the employees and how they can try to avoid it as much as possible. In addition to organization to develop plans and terms to try to avoid the ICT and to try to impose a new strategy that will help the employer productivity in the space and in the workspace. Lastly, is that this paper is one of the rare papers that investigated in the developed economy. In addition, we applied the MGA analysis that was not mentioned here due to the limitation and the time constraint. We also believe that our paper will help the higher education management to take into consideration the negative aspect of the ICT use and how they can avoid it and try to improve it in a way that will allow the employee to be more productive in their workplace. As we can see today, due to the COVID, technically every individual and in every home there is an ICT, and a lot of them depending on it to do the daily life and daily tasks. And we can see that the understanding of the ICT is really important, so the employer does not feel that they are it's a hindering way to achieve what they need to do. Despite all of these, we believe also that our paper, if we manage to extend it, we would like to extend it to other sectors, schools and business firms, as everyone will be different from one to another. And inshallah, maybe we believe that we can incorporate it to other countries. Lastly, I would like to thank QNRF for supporting us in this presentation, allow us to be part of this opportunity. A deep thanks for our professor, Dr. Shada and Dr. Allam, for supporting us during the past 2 years of this whole process, and also for our Qatar University for allowing us to be participating in it, in addition to allow us to participate in one of the international conference in India, where we won the Young Scholar Relationship research paper. Thank you so much for your time, and I'm open for questions.
Thank you, Ala. You will receive the first question from Dr. Hisham Saber.
Hello, Ala, and thank you very much for the presentation. Your presentation, or study rather, concentrated on the effect of use of ICT by employees on various criteria. To what extent the study could be extended to study the effect of how much use of ICT can support employees rather, for example, we currently holding this virtually, and this is something that we couldn't do without ICT support. Exactly. At QNRF, for example, we are a paperless organization, and we rely on ICT in support of various functions. Mm-hmm. It allows remote working from home, flexible hours, and so forth. So to what extent the use of ICT in support of employees rather than being used by employees would have a positive effect rather than a largely negative effect. Thank you.
Okay. So we do understand that the positive effect of the ICT is open and everyone is depending on it because that's where we want to achieve. And as we can see nowadays, the ICT is in everyone's life. However, what we're trying to find is, despite that it is allow us to be virtual and we do conferences and online, the community of the work life of the work employee or the organization, let's say, it gives the employee, it added pressure on them that they have to be connected. So for example, I finished my working hours, but then at 6 p.m., I know I have another meeting with my team colleagues because since you're home, you have to be connected to 24/7. So there is a positive effect, and then we understand that it is a very impact us, and it allows us to be much open to a lot of research and to see the benefit of it. But despite this positiveness, the negative is also part of it, because to be connected 24 hours as an employer, even as an organization, I'm still affecting the after working hours, affecting myself, affecting the society around me, instead of trying to balance myself to be able to give the society and at the same time give myself as an employee. I hope I answered to what you're expecting.
You did. Thanks very much.
Thank you, Dr. Hisham. The next is Dr. Hatem. Please go ahead.
Thank you, Ala, for this presentation. I think it's a very interesting topic, and I have a couple of questions, rapid. The first one is, in terms of definition of concept, what is exactly the difference between ICT use and ICT demand for you?
Okay. So the ICT use is the use of technology. So such as phones, laptops, connecting to different platforms. This is the use of the technology. I'm using technology to access all of these.
So the question in your questionnaire was exactly, do you use your mobile phone or your laptop one time per day?
Yes, during your life or during your work, what type of technology you're using to approach the ICT. And the ICT demand is basically the belief I'm expecting my employee to be connected to those uses of technology and having expectation that they have to be connected 24/7 to reply to these. And I'm having also the mindset that they know how to use it. So I'm putting a pressure on them that, okay, you can use technology, I'm assuming that, and you can be productive anywhere, so because you have to be connected to it.
Okay, thank you. Your sample is constituted by more than 80 observations, no?
Yes, the sample was 100 and it was distributed in Qatar University on different academic, non-academic stuff to answer the questionnaire. And then it was collected after a period of time.
So why you use the PLS and not the traditional regression econometric models like OLS?
Because the PLS is more accurate and more researchers would recommend to use PLS in these type of models and analysis. So we went to use the PLS in terms of a new way. That's a new way, even different from what we study in university. And we wanted to test, will this actually be accurate using the PLS, or we should use the general Excel and regression as you mentioned. However, when we see the MLS, we found that it's accurate, it's more reliable. And also, as I mentioned in the research, we use the MGA analysis, which is the multiple group analysis, just to confirm that is the PLS accurate or not. So we tested the MGA analysis by having the same sample and then subsample it into 3 groups again to ensure that each sample were accurate. It did give an indication that yes, the sample was correct, and it confirmed the PLS analysis.
Okay, last remark.
Yes.
Did you divide your sample, for example, between man and woman to see if the results change or not, between academics and administrative to see if they have their own specificity.
Different. Yes, I believe this question is really, thank you for asking this, Professor. We did distribute it on different segments, female, males, academic, non-academic. But I think it can be considered as a limitation because maybe it didn't show in the presentation how the female will have the effect of negative aspects since the life is different, how the men will have it. I think this could be a good addition to add in the presentation, and we will consider it. But in terms of analysis and the sample, they were included as well.
Thank you, Dr. Hatem. I think the time is allowing us just for one more question coming from Dr. Amar.
Thank you, Hala. Good morning, and thanks for the presentation. Hala, my question is on the same lines of Dr. Hatem and Dr. Hisham. While I understand outside working hours, the employee might feel obligated to or obliged to stay online, etc., and that puts some stress, and that's why they are not satisfied. But within working hours, the results are surprising, at least to me, is why wouldn't they be satisfied to use ICT within working hours? So the question is, did you subdivide your analysis between within working hours and outside working hours? And if so, how do you explain the unsatisfaction or the dissatisfaction within working hours?
Well, in terms of this, I believe we kind of missed it. I do admit that how is it during the working hours, but we focus mainly on the after working hours. But to answer to your question, and based on the research that we did, and based up to my knowledge, during the working hours, they might not be satisfied in terms that the company or the organization, they will ask the employee to use a specific technology. For example, when we started using Zoom, many employees will not be able to know how to connect to it, and there was a lot of technical issue. But were there supported from the organizations themselves to support this employee to use it in the correct way, or to show them, okay, if Zoom is not working, there's other platform that you can use and more easily to use it, taking into consideration not everyone is the same age. So for my example, I can be easily understanding the ICT and I can adapt to it and easily working with it. But for older age group of employees that could be 15 and above, they feel stressed. They feel that I'm trying to understand this technology, but it's not working with me. So what I'm supposed to do? And that lead them to think that, okay, within working hours, I'm trying to connect, but in this after working hours, it's still affecting me because I'm trying to understand it even after working hours. So I can be able to use it during the working hours. I hope I understood what you meant.
Yeah, thank you.
Yeah, thank you, Dr. Amar. Thank you, Ala.
Thank you so much. Thank you.
Thank you very much. And the last in the social science is a project from Georgetown University by Mohammed Al-Khleifi, Osman Kamara, and Saoud Al-Ahmed. And the research title is Jidariya. Please go ahead.
All right, good morning, everyone. We'll try to keep this portion of the presentation quite short as we want to maximize the time that we have towards the end of the presentation to showcase our documentary. All right. So the scope of this research begins following the blockade of Qatar on the 5th of June, 2017. And when we talk about a siege on a nation from neighboring countries, this would leave a great deal of panic, uncertainty, and internal turmoil in the country. However, what is interesting is that in Qatar's case, the exact opposite ensued. And there was a stark increase in nationalism and solidarity across all facets of the society. And you have Qatari nationals, expats, blue and white collar workers. All of these people came together in an unprecedented manner in order to celebrate this symbol that arose from the major event, and that is the Tamim al-Majd Jidariya, which features a painting done by Ahmed al-Ma'adi. In the past, when you have like such display of adulation for the emir, and the nation was only limited to events such as the annual Qatar National Day celebration. And it was more or less a spectacle. So following the blockade, it became clear that Qatar nationalism, Qatar solidarity became visually represented by Tamim al-Majd billboards, not to mention car stickers, mugs, t-shirts, and badges, all these things. And it was done constantly and year-round, and even till today. So the messages written on the Tamim al-Majd billboard serves as a significant insight into how these messages, regardless of the language of the writer, all showed a sense of belonging that stretched out of the Qatari national sphere into the sphere of that of the migrant communities and the expats. And we find that while the issue of the ongoing GCC crisis has been discussed extensively in English and Arabic language news media, there has been a lack of studies that analyze the unique and unprecedented proliferation of the visual communication of Qatari nationalism in the form of these murals, the billboards, the posters, and the drawings. So basically, this project aims to gain a better understanding of how political crisis can be translated into a visual phenomenon through social participation. So given this, anthropological theories and methodology were pivotal in helping us reach that goal, and really tackling the points of interest in visual studies and sociolinguistics. So this is something that Uthman will further elaborate on.
Hi. Can you hear me?
Yes, go ahead.
Can you see my presentation?
Just enlarge.
Osman, as we discussed, you need to go to the presenting mode. No, the icon before. Where
this one?
Down there, yes.
This one. Yes.
Okay.
And
So
please use a laser pointer as well.
Can you hear me?
Yes, we can.
Yes.
Okay, to continue from where my colleague stopped. So as we are all students and from different majors at Georgetown, I think some of the methods we try to use in order to come up with effective effective research was we try to organize ourselves and give each and every student a task, because we have to balance it with our classes too. So some of the things we did, we try to do an intensive background research for the resources we're going to use during the research. Some of the things are like the equipment, the cameras, and the articles and the newspapers we have read in order to try to come up with a good research result. So and some of the research methods also which we really needed, and they're effective for any research where qualitative methods and quantitative methods. So in qualitative methods, we invited people like students, those who are internationals and locals from Georgetown and outside Georgetown to interview them, and we scheduled time with them as students, to interview them about the project itself, about the Jidariat project. And the quantitative method we use, also was, like I mentioned at the beginning, we familiarize ourselves with the readings, of our project and some of the equipment we should use. And we, as the students, we were the one who designed the questions, what kind of questions to ask the locals and internationals, by using the, and we've used another method also, visual anthropologies like images and documentaries. And at Georgetown, we interviewed some students there, and film footages also. The film footages we took, it was, compartmentalized into different categories, and we edited with a Premiere Pro. So in order for us, why we choose this methodology, because we think that this is the best methodology for us as a student in order, to do well in our research, and whilst it will not affect, our studies also. And we have taken pictures throughout, Qatar, everywhere in Qatar, in different locations in Doha, in Qatar, and some of the limitations we had, because some of the the images and videos we couldn't use, for certain reason because the qualities of some of the videos. And secondly, also, I think one thing also we have done better, was the systematic way of our research. We try to meet every 2-3 weeks to discuss and to bring in the materials we have and the ones we should use and the ones we shouldn't use. So yeah, this was part of our methodology we use. Yeah. So Saud will continue from there. Hello.
Hello, everyone. Hi. I'm trying to share screen, but Athman. Athman, can you stop the screen share?
Okay. Stop the screen share so he can open his.
Thank you. So the results of the major result of this documentary was the tangible visual anthropology piece, or the documentary based on the interviews with the students. As Athman said, the students the interviewees come from a variety of backgrounds: international, local residents, Qatari nationals. And this is just to show how there's a combined spirit towards the image or the spectacle, as Mohammed Al-Khlaifi put it, towards the Jidariya or the image of Sheikh Tamim. Other than the visual tangible piece, this project also honed and polished students' research skills, and that's because we learned how we almost gained a new level of research, because it was not only simply coming up with questions and going to interview people. There were many stages to forming this research. We looked at as Athman put it, we went around, looked for Jidariyas, we filmed them. We looked at the word.
Saud, excuse me, Saud, you have 2 minutes left.
Okay. We looked at what people said and what they, like the words they used, and that's what we also studied. Okay, I'm gonna move quickly because of time. Conclusions. For limitations, I'd say our limitations was, even though at the time of filming, the Jidariyas were decreasing or being removed because they were being filled up, but the spirit of the Jidariya was still there. People still had a sense of nationalism in them. And post-project plans, quickly, we are planning to submit this project to publish it on Camera Obscura, and we're still working with the professor for that. Acknowledgments, we acknowledge the Professor Abu Sharaf and Professor Yahya for their effort in helping us with this project. And special thanks to Muhammad Al-Jabri, who had a pivotal role in this project but couldn't make it today. As for the documentary, I want to show a few minutes. So the question here was, what does the Jidariya mean to you?
بعد الحصار أصبحت دولة قطر معتمدة على نفسها أكثر. I look at it as an artistic way to show the people's support to their leadership. So it's an incorporation between art and loyalty. That's how I see it.
First time I wrote on one of the Jidariyat was the one in my city, next to Hilal area. I wrote, we are one, or we are Tamim. Say that, Killina Tamim. I wrote that because I felt like that's what the photo was about. That's what the painting was about. It was about Sheikh Tamim and how he helped us unite. He helped us set that stand as one.
The Tamim al-Mas poster, like it to me symbolizes Qatar. Makes sense. When it all started, they had a very big one put next to our house in Abu Hamur, and so I had to go out and sign because I felt obliged to do so. And when I went there, the first time I went there, I remember signing in Arabic, Killina Tamim al-Majd. Obviously, that became a mainstream thing. Everyone did the same thing. But then I took it upon myself to go to other places located in Doha where they had the Jidariya put up, and they had like cranes and whatever to lift you up because there wasn't enough space. And so I kept on writing like in Arabic or English supporting the royal family. And even in Georgetown, they bought one, they put it up there. I went there and I wrote that we all support Qatar, we all support the Emir. And it was also interesting to see how many foreigners interacted with this. And they've also shown their love for our Sheikh, which was interesting to see. And it really showed the unity of not only Qataris, but the expats, the foreigners, because they felt safe at home.
Okay.
Yeah.
We'd love to receive the first from Dr. Hatton, please.
First
Yes,
of
Dr.
all,
Hatton.
I want to congratulate with you for this very important work, also for the smart idea, I think. But at the same time, I will be frank with you. I am somehow frustrated because I feel that the central person in this work is missing. Do you understand me?
Yes, the person behind the painting.
I agree with you. I feel like he has an important role in shaping this image, but it's not so much about the creation of this image. It's more about how the image became a symbol or a spectacle or a manifestation of people's nationalism. There were millions of Tamim, or not millions, sorry, but there were many drawings and art pieces that have to do with the Tamim al-Majd that came out. But this one just happened to become a symbol and a manifestation, and the most widespread one. Arguably, it could, I argue, Yani, that it just caught on with people, like, it caught on with the spirit of the spirit of what's going on, and it's a representation.
Okay, have you tried to contact the cabinet of His Highness to have an interview with him?
With His Highness Sheikh Tamim?
Yes.
No, we didn't.
You bring up an interesting point. This is something that would definitely act as an extension to our research. And to add to your previous question, yes, we also did, in case you were wondering, reach out to Ahmed al-Mu'alid, the person behind the Tamim al-Majd painting that first came out after the blockade. However, it was not so much so the significance of the artist. Of course, we're not denying that there was a huge significance of the artist in producing such a significant work that really resonated across all facets of society. However, our project really aims to gain a better understanding of how a political created into this visual phenomenon through social participation, through the participation of the different members of society, going up to the Jidariyat and participating by writing their messages. However, yes, we do understand where you're coming from. It would have been a core addition to this project.
Yes. Suppose now that you have you've had this great opportunity to contact His Highness to have an interview with him. So, what are the main questions you would ask?
So this is to His Highness.
Yeah, if this was to His Highness, one question I had, like while we were interviewing and while we were asking people, was that what would he think about his image being everywhere? Like, what would because it's a strange thing having your face everywhere, like on billboards, on I didn't show this part of the interview, but we asked what was the strangest place you saw the art piece of Sheikh Tamim. So I would ask, what do you think of having your face be a symbol of resilience and nationalism, because it's not very orthodox in a way.
Thank you.
Thank you. Just one point to clarify to all audience. I'm just pointing at when I asked Dr. Hatem to start, because this topic is relevant to his function and role at QNRS. So that's why I'm allowing him to give more questions on the subject. So the next is Dr. Jarrar, please go ahead.
Yeah, thank you for this nice work. I have a question. The power of visualization to reflect the social opinion, I think it should include more diverse individuals. Why you limited your interviews and the people you show here to students and young age or educated in a sense? Did you think that will be more powerful if you included more different sectors of the society?
Well, the process is, it's not, I mean, it's not really as easy as going up to people and, you know, just inviting them to do interviews. When we took our IRB training, we wanted to ensure that we didn't come across as people exerting pressure on others. Therefore, you know, those who participated knew that there was no pressure. We wanted to make that very clear to them. And there are, of course, consent forms that were involved. Would we want more people involved into this project? Definitely. This is something that would enhance our results. However, I mean, given the time frame that we had, I would say that overall it was a successful endeavor. However, yes, you do mention an important point. Would be definitely an important addition to enhance our research.
Okay, thank you.
Next, Dr. Drew, please.
Yeah, thank you very much, and very nice presentation and a very interesting piece of work. A question for you: did you actually consider looking into the social media as well, or was this the physical billboards that you looked at?
We looked at social media, but in the sense of the words people would use, like the linguistics, and that's the specialty or where Professor Yahya Muhammad comes in. He's a linguist, and he like helped us research the wordings of things and how people put things, like كلنا تميم, sayings for national days, like مطوعين الصعايب, their significations and things. So that's where social media really came in, and tracing the origins and the meanings of these nationalistic phrases.
Thank you. Yeah.
Thank you. The last question is from Dr. Hisham. Thank you.
Hello, gentlemen, and many thanks for a very topical project and presentation. The one thing that wasn't clear to me from the presentation, although I'm almost certain that it is there in the project, is the academic content. You got a lot of input data in terms of interviews and so forth, but what did you do with them? Did you analyze the content of what people saying, the relation of that to their various backgrounds, and the implication of that and so forth? Or was it just a matter of just producing a short footage?
The short footage was a byproduct of the research that we did. However, we definitely did use some methodology that perhaps maybe Athman could further elaborate on. But just to give a brief input on an attempt to answer your question, there was visual anthropological methods that was used, and it really helped us kind of map out the nationalism and the migrant and host dynamics involved with these with the kind of the influx information that we got from these interviews. And it's a process that took time, and we had to really sit down with our mentors. Professor Ruqayya Boucharaf, many thanks to her, as well as Professor Yahya Mohammed, who kind of gave us the necessary material, the readings, the methodology to be able to kind of circumnavigate through these interviews. It's not as easy as just taking the interview material, putting it together in a video, and presenting it. But there's a lot that actually went behind conducting the interviews themselves, producing the necessary questions that really kind of go at the core of this Jadariya project, and at this social participation that we see following the blockade.
Thank you, team from Georgetown. I think that was excellent presentation, as you could have managed 3 presenters to do 1 presentation. I think this is a good effort. Yes, now we have come to the end of the 12th annual UREP competition. And before we signing off, I want to congratulate all our student researchers who participated and presented their exciting research projects in this competition. Moreover, I would like to thank our distinguished judges who now have to make the decisions for the winning presentations. Decisions will be announced very soon on our website, so stay tuned for updates. I would like to thank our team, and the most important team in this competition is the effort of Mr. Wael, who designed the platform and made an easy access to this platform. Sheikh Abdullah, the PR manager, for his contribution to disseminate the event internally and externally, and to Mira Asleti, who is helping us as intern for the effort in communication with the students and the follow-up to provide their presentation and their posters. So I would like also to thank my team, the CB team, capacity building team, Noha and Dr. Ayman. And lastly, thank you all to all those who joined us today. And I wish you all health and happiness. Be safe. Stay safe and goodbye. Thank you.
Thank you.
Thank you.
Thank you.